Spectrophotometry is a very useful tool that we have in the field of biology as well as chemistry, and it deals with how different substances absorb light. So different molecules absorb different wavelengths of light. Not all molecules will absorb all wavelengths of light. They tend to absorb different wavelengths differently. And we can use that quantity of light that's being absorbed by a substance to then determine the concentration of that substance in solution. And that's what today's video is about and how we're going to use it in lab. So as I mentioned, substances that absorb light tend to only absorb in certain wavelengths, not all wavelengths equally. If we look at the absorption spectrum for chlorophyll A and B, what we see here on the x-axis is the wavelength or color of light going from the blue end to the red end, and on the y-axis is how much of each wavelength is being absorbed by that substance, by chlorophyll A or by chlorophyll B. And as we move from left to right here, we can see that chlorophyll B absorbs best in the blue wavelengths. It also absorbs a little bit down here as we get to the orangey reds, but it absorbs best here. Chlorophyll A, on the other hand, does absorb in the blue, but it really does its best absorbing at wavelengths in the red. Neither one of these two pigments absorbs very well at all in the green middle of the spectrum here. So right away we can tell, we can develop a hypothesis from looking at this absorption spectrum and say that plants will undergo photosynthesis best either under red light or blue light, but they won't do much photosynthesis under green light because that green light isn't being absorbed. That energy isn't being absorbed by the leaves. Instead, it's being reflected. That's why we see leaves as being green. So looking closer at how a spectrophotometer, how the actual machine works, here are the main pieces that we're going to be concerned with. You're going to have a source of light, and then you're going to need to select what wavelength. Remember that lambda is the symbol for wavelength or color, so that we can select exactly what color of light we want to pass through our sample. On the opposite side of our sample, then, we have the sensor, and that's going to register how much light actually made it through our sample. So the first step in using a spectrophotometer is blanking the spectrophotometer or zeroing it out. And to do this, we use a vial that's square or cubic, that's referred to as a cuvette. So that's the cuvette, is the vial that contains our sample. And in that cuvette for our blank, we want to just introduce solvent. Solvents themselves will absorb certain wavelengths of light, and so we need to make sure that if our solvent is absorbing any wavelengths of light, that we take that into account before we measure our substance of interest. So it's very much like tearing a balance. So when you start to weigh something, you put the weigh boat on the balance and then you press tear to zero out that balance, so that when you actually weigh your chemical, you know you're only actually weighing the chemical, not the weight of the weigh boat and the chemical. The same idea here. We don't want to include the absorbance of the solvent. So our blank is just going to be filled with solvent, and the sensor is going to measure how much of this light makes it through the solvent and is recorded by the sensor, and then zero out the spectrophotometer so that we've already taken into account that. Step 2, we can introduce our solution, and in this case our solution is what we ultimately want to know the concentration of. And as I've tried to diagram here, certain solute particles will absorb photons of light in preventing them from reaching the sensor. So you'll notice that less light has reached the sensor because some of the photons have been absorbed by our solute. If we increase the concentration, more photons will be absorbed and even fewer will reach the sensor. That's the whole concept of relating concentration to light absorption. So Beer and Lambert, Lambert often gets left out of this, but Beer and Lambert developed a mathematical way of relating how much light is absorbed at a specific wavelength to the concentration of the substance in solution. And when we look at that mathematical equation, it's absorbance equals the extinction coefficient, which we'll talk about in a moment, times the distance that the light travels through the substance. Now, for most spectrophotometers, for most machines, that light path has been built in as being 1 centimeter, and we'll see why that is handy later on. Times the concentration of your solute, and that can sometimes be in milligrams per milliliter. Sometimes there will be other units, and I'll talk about that in a moment also. So if we looked at a curve of concentration versus absorbance, what you'll notice is that as concentration increases, absorbance increases in a linear relationship, at least for a while. At some point, that curve will level off, and any concentration greater than that point, you won't be able to determine using spectrophotometry, because even though concentration is still increasing, absorbance isn't increasing anymore. So you've lost that relationship. So you can only really use the linear portion of the curve to determine concentration. So extinction coefficients, as I mentioned before, can be expressed in different units, and that's largely dependent on the discipline or the substance that's being studied. For chemistry, it's very common to have what's called a molar extinction coefficient, which is the inverse of moles per liter times the inverse of centimeters. Those would be the units of the extinction coefficient. In biology, in certain areas, it's common to see the extinction coefficient expressed in units of inverse milligrams per milliliter times inverse centimeters. And that's what we'll be using to determine chlorophyll concentration, although in a future experiment. But we will also use this first measurement as well. And I'll talk more about that when we get to our chloroplast experiment later this semester. Another thing to know about extinction coefficients is that they are very specific to a given wavelength. So when you look up the extinction coefficient for a given substance, you also need to know what wavelength was used to derive that extinction coefficient, and it's also influenced to some extent by pH. So you'll need to know the pH at which that extinction coefficient was measured. For our purposes, it's most common to have pH of 7 or very close to 7 when determining the extinction coefficients, but it's something to be aware of. So here's our Beer-Lambert equation. Absorbance equals extinction coefficient times the path length times the concentration. We're going to measure the absorbance. The extinction coefficient for chlorophyll, and actually this is the extinction coefficient for a mixture of chlorophyll A and B, is 34.5 inverse milligrams per milliliters inverse centimeters. As I mentioned before, it's pretty standard for spectrophotometers to have a path length of 1 centimeter. That's the distance that the light travels through your sample. And ultimately, we want to find out the concentration of our sample. So we solve for C. You rearrange this equation, and it becomes absorbance over the extinction coefficient times the length. And because that's 1 centimeter, that length becomes 1, so kind of just can ignore it. It becomes A divided by 34.5, gives you your concentration, and because these were the units of the extinction coefficient, your concentration will be in milligrams per milliliter. The centimeters will cancel out, and you wind up with just your concentration in milligrams per milliliter.